Oh. Hello, welcome people on Zoom. I'm just trying to figure out how to get the slides to actually go up on the board. Okay. So we're starting off with a new unit today. We're going to do a very brief overview of the last stuff in chapter 9, which I know you already did the homework in last unit. I think we got like 5 slides on that. Overall, you guys did great on the exam. Super pleased. Most of you, that went really well. Brief reminder, you do want to withdraw from this course. The deadline is today. So if that happens to be what you plan to do, shoot me an email today so that we can get that through. Yeah, there is another section of A&P1 in the spring, so you would have time to catch up if you feel like you're too behind at this point. But I'm going to assume that you guys are planning to stay. So let's roll with a little bit on the CNS, the central nervous system. Yeah, and I put it in the folder, just these new like condensed slides on set exam 3. So you should find it there. Yeah. It's basically like one slide on microglia and then a reminder that the blood-brain barrier exists, which I'll talk about what that is. Yeah. That's what we want to know going forward. So in our last unit, what we were talking about was the neurons, but the central nervous system does include other things. So the central nervous system, as you saw in lab, has your brain and spinal cord. And then we have the peripheral nervous system coming out of that, which is what we'll mainly be talking about today as we move into the autonomic and somatic nervous system. But the central nervous system is super important. It's what's maintaining homeostasis. We have 100 billion neurons and 100 trillion synapses. And now you know everything about how they all work, right? Having taken exams too. So. In the central nervous system and out in the rest of the body, we consider 2 things to be nervous tissue. One is those neurons that we talked about endlessly. So those are the ones that conduct our impulses. And a neat thing about neurons is that they generally can't divide. So you're kind of set with the number of neurons that you have, the number of nerves you have. But cells that can divide in the nervous system are these supportive cells called glial cells, or neuroglia, if we want to emphasize that they're part of the nervous system. So they have supportive jobs in the nervous system, but they don't fire action potentials. They don't do any of those things that we were talking about in unit 2. However, they can divide. And some of them are implicated in various disease processes, which is why I want to just give you guys a quick reminder that they exist before we move forward. Because you may someday in the future, as you're learning about various diseases, want to come back to this. I think you should have seen this more or less in Phys Lab. But when we're talking about neurons, we can classify them. One way is based on shape. But the way we care about for this class is functional classification. So we have neurons that convey senses. So we can call them sensory neurons or afferent neurons. We'll call them a lot in the future. and they conduct impulses out from sensory receptors to the central nervous system. So when you touch something and you know you touched it, that's because you have sensory nerves sensing that pressure. You lick something and you can taste it, you have sensory nerves for that as well. We have all kinds of sensory nerves, afferent nerves, bringing that data about the world into our central nervous system so we can process it. Motor neurons, which are a type of efferent neuron. So efferent means going out. They conduct impulses from the central nervous system, so our action potentials, out to target organs, which are going to be muscles or glands. Muscles and glands, really the bulk of this unit, other than the divisions of our nervous system. Finally, we have association or interneurons. They're located in the CNS, and their job is just to integrate within various parts of the nervous system. So they don't go out and do stuff in the body, and they don't directly sense anything. So they're kind of an in-between. Our glial cells actually compose most of the CNS, which is why I don't want to skip them entirely. And we have 5 types that we want to know about. So the first are our astrocytes. Astro means stars. So that's these big green ones with all these branches. And their main job, although they have many jobs, is to create this blood-brain barrier, which is basically to kind of like seal up holes in the capillaries in this blood flow through the central nervous system, so like through the brain, so that a lot of substances that could usually leach out into your body can't leach into your brain. And we have ependymal cells. Their main job is to line cavities, and by cavities, we really mean those ventricles in the brain that you saw in lab. So they're the ones that are making CSF, the cerebrospinal fluid. Our microglia are kind of like the immune system of the brain. So they protect the central nervous system from foreign matter from different particles through phagocytosis, which is just like eating something, right? So they surround things, pull them in, and break them down. Then we have 2 types of neuroglia that form myelin. So we talked about myelin. It's that sheath that forms around some axons that speeds up conduction. It speeds the ability of action potentials to race down those axons. And in the CNS, it's formed by oligodendrocytes. And in the PNS, it's formed by Schwann cells. In this picture, I pointed out the astrocytes, the green ones. These orange ones around the ventricle are the ependymal cells, where we have microglia. Microglia are these purple spidery ones. We can see them not directly really touching anything because they're wandering around looking for bugs to eat, right? And we have our oligodendrocytes, are these purple ones that branch out to many neurons to form myelin. And then out in the PNS, we have Schwann cells that just wrap around individual kind of neurons or nerves. So the important thing about the astrocytes, and one of the important things about chapter 9 that I don't want to skip, has to do with blood flow in the brain. So you looked at the pathways of blood in the brain in lab, but what we're going to care about here in the lecture is that the brain actually uses a ton of oxygen and a ton of glucose compared to how much of your body it actually composes. Hopefully you all got to hold that brain in lab. It's, you know, maybe 3 pounds, something like that. It's consuming 20% of your oxygen and 50% of your glucose, right? So like half the glucose in your body is going to keep your brain functioning. Yeah. So what we want to know about the nervous tissue is that neurons, they depend on aerobic glycolysis, right? So they require oxygen, and they're actually going to require glucose. They're not going to be able to use very many alternate sources for energy at all. So we talked briefly at the end of our metabolism unit how you could break down proteins, how you could break down fat in various places to also make ATP. A neuron can't do that. A neuron really needs glucose. It needs glucose and it needs oxygen. So you're not going to have like lactic acid forming in your brain. We're not doing anaerobic stuff in the brain. So we need to make sure we have blood flow, and we need to make sure we have glucose and oxygen going to the brain. If we don't have blood supply and therefore don't have glucose or oxygen going to the brain, we have issues. A stroke is an example of that. So a stroke is really decreased blood supply to the brain, either because a blood vessel has been blocked. So occlusion means like a blockage. So if you have like a blood clot, block off one of those arteries you looked at, that could cause a stroke. The other thing that could happen is you could have hemorrhage. So you could cut a blood vessel or burst a blood vessel. An aneurysm, it is like a burst of a little bit of a blood vessel in your brain. So that would also cause a stroke. Blood-brain barrier. We will talk about capillaries extensively in the future. I know we haven't talked about them yet, but the structure of a capillary is basically, we have these endothelial cells surrounding our blood, making a tube. And there are little places where different types of molecules can go through. So some things are going to cross through these cells, but there are also pores where things that can dissolve in water, so anything that's hydrophilic, can communicate between our interstitial fluid and the blood. So we do have the ability of a fair number of things to move between our blood and the tissue surrounding. But we want to really tightly control what can get into contact with our nervous tissue in the brain. We really want to control what can touch the central nervous system. So in the brain, we have this special anatomy where we pull those cells closer together and we kind of block them up with the astrocytes, those neuroglia, those glial cells. And this means that we're going to kind of block these pores. Are actually going to form some tight junctions here. And this is going to prevent things from moving between our cells. And the only way things are going to get to cross is if they can go through these cells forming the wall of the blood vessel. So this just gives your body a bit more ability to control what's moving into the tissue of the brain itself. So we can see here, they've specified this blood vessel is touching cerebrospinal fluid, which is that fluid that bathes your brain and your spinal cord. Finally, myelin. So really the only thing I want you to know here is that you have different cell types making the myelin in the central nervous system versus the PNS, the peripheral nervous system. So just pure word association between oligodendrocyte, CNS, and myelin will get you where we're going. So an oligodendrocyte basically creates myelin on multiple axons, so on multiple nerves. Myelin in the PNS, so out in your body, you're looking at the brachial plexus in lab, so those big nerves that you see in your arm, they still have myelin around them, but it's not made by oligodendrocytes. It's made by Schwann cells. And it's going to look a little different. So we notice how those nerves are separate from each other, right? And part of that is that they have myelin surrounding those individual fibers, right? And that's our Schwann cells. So a Schwann cell is like a pancake that wraps around and around and around a neuron in the PNS. And we can note here, because these are words that are out in the world, you may have heard about white matter and gray matter. So when we're talking about myelin, that is what white matter is. The myelin itself is white. So white matter of the brain would be found kind of in the central part of the brain if you cut through. So that would be full of all those connections, those myelinated axons. That's why the corpus callosum that you saw in lab was lighter in color. We have tons of myelinated axons running through there. And gray matter is unmyelinated stuff. So primarily that's going to be cell bodies and dendrites, because the myelin is on the axons. Questions about those microglia and those very few topics on the CNS? Hopefully that's fairly straightforward. In that case, we're going to move into the autonomic nervous system. We're going to start by describing its anatomy and kind of some of the divisions of the nervous system. On this slide, we have a map of what's going on. So what we've talked about just now, the central nervous system and the brain and spinal cord, can be then split out as we get into the peripheral nervous system. So we start to have these divisions. So we can see that these are actually arrows. So what we see here is that we have receptors out in the body, bringing in sensory information through the peripheral nervous system. So this is afferent or sensory fibers are transmitting information out from those receptors, which we call visceral receptors. They're visceral specifically if they're in sensory organs. There are some other types of afferent fibers going to other types of sensory organs, but the difference is not important for us here. And it's important that we can sense what's going on, because otherwise we can't maintain homeostasis if we don't know what has changed, right? So afferent fibers are important in maintaining homeostasis. So that's in the peripheral nervous system. We come into the central nervous system, and then it's where we branch out back into the peripheral nervous system with our efferent fibers that we start to have these divisions that we're going to talk about over the next today and probably on Wednesday as well, because we have Monday off. So we start to have these efferent fibers with motor commands going out of the central nervous system and into the peripheral nervous system. Then we're going to classify them more specifically. So our division first in the peripheral nervous system, we're going to have what we call our somatic nervous system and our autonomic nervous system. So the somatic nervous system, the one that's going to your muscles, right? So that voluntary stuff that you move, like a soma with a cell body, somatic is going out to like the physical part of your body that you think of, right? That movement that you interact with. The autonomic nervous system, we can think of a more kind of like involuntary kind of nervous system. So your autonomic nervous system is going to be acting on glands, which we'll talk about with chapters 6 and 21 for our endocrine system. It's also going to be going to the two types of muscle you can't control. So to your heart and to smooth muscle, which we'll see in the future lines things like your blood vessels. You can't really intentionally contract a blood vessel. So that's going to be supplied by the autonomic nervous system. So anything that you're not thinking of as being under conscious control, I want you to think about the autonomic nervous system. Then the autonomic nervous system itself has a division. And our two parts of the autonomic nervous system we talk about are the sympathetic and the parasympathetic nervous system. So as we move through this, we're going to think about the sympathetic nervous system as being your like fight or flight response. So basically anything that stresses you out is causing a sympathetic response. And your parasympathetic nervous system is going to be the rest and digest. It's going to be controlling nice, relaxed kind of thoughts. So before we move forward from this chart, I know it can be a lot to kind of think about all the different ways we can classify these parts of the nervous system. So any questions on this before we move forward and start getting into the nitty-gritty of sympathetics, parasympathetics, receptors with sympathetics and parasympathetics, et cetera, et cetera? Yeah, and you can come back to the map later. Just we're gonna be diving into the details relatively quickly here. Okay. But at any point you start to feel lost, come back and zoom out to this diagram. Okay. When we're talking about the autonomic nervous system, we want to be aware that our two divisions, the sympathetic and parasympathetic, they're usually going to the same place. So like, if you think about your heart, it can speed up, but it can also slow down, right? So that's why it gets both divisions, the sympathetic and the parasympathetic, because they're basically causing opposite responses in the same structure. So that's why we say that they have dual innervation, because they have like two types of nerves going to them that are going to kind of control where you are on sort of a sliding scale. We'll often see that both parts are even active. It's not like a light switch, it's kind of like a dimmer switch. Whether you're closer, you have more of one, or a little less of the other. So your autonomic nervous system is operating in relative secrecy, meaning that like you hopefully don't notice it, right? It's doing its own thing kind of on an involuntary basis, kind of an automatic process with this autonomic nervous system. The parasympathetic nervous system is going to be doing the resting part. So like that would slow your heart down, for example. The sympathetic nervous system, stressing you out. This is a fight or flight response. So it would, for example, speed your heart up. I'm just using the heart as an example because I think it's an easy kind of touchstone for us, but each of these would also be having effects on glands, also having effects on our blood vessels, but the same kind of principle applies, that we have changes in opposite directions depending which part of the autonomic nervous system we're talking about. So there are lots of unconscious processes, like the beating of your heart, that are going to be controlled by these two systems. And it's important for maintaining homeostasis, like your heartbeat, right? Your heart rate partially is controlling blood pressure. Right? We'll see that these systems also help you control blood volume. There are a lot of unconscious processes, things that need to be maintained by these systems. Okay. So ANS is an abbreviation for the autonomic nervous system, which is a part of the peripheral nervous system. Lots of acronyms. The autonomic nervous system, really important for homeostasis. So here we see a big map. And for right now, I'm just going to point out, so our sympathetic is in green on the left. Our parasympathetics are in purple on the right. You'll notice here I was talking about the heart, right? It's receiving both green and purple. Lungs receiving both green and purple. All these organ systems in the center are receiving branches from both sides, basically. So we have dual innervation. But you'll see this green part on the right, or sorry, on the left versus this purple part on the right. They're coming from different places. We do see that there are some different intermediate structures here between the two. So there are differences in the way these two nervous systems work, even though they're going to the same places. So, like when we were talking about neurons, when we talk about these divisions of the autonomic nervous system, we're going to talk about preganglionic neurons and postganglionic neurons. So before we were seeing presynaptic and postsynaptic. Now we're calling them preganglionic and postganglionic. But it's a similar concept. It's just that we're not zooming on a single synapse, right? So if we looked on this image, right, we have a synapse here, we have a synapse here, we have a synapse here. And now we're zooming out a little and we're looking at the overall relationship between these two cells. So the CNS is going to send out a preganglionic neuron to what we call an autonomic ganglion. So ganglion is just going to be a place where you see cell bodies, basically. When you see them in lab, you may actually be able to pinch them. They can actually get large enough to see. So we might have collections of cell bodies together. And this is where we're going to see the communication happening between that preganglionic neuron coming out of the central nervous system and the postganglionic neuron that's going to our organs. So we call them effector organs. We actually saw this term way back, probably on day one, definitely in unit one. But our effector organs are those organs that actually have an effect for maintaining homeostasis. Right? They're the ones that are actually doing the action that modifies the body in such a way we can get back to our set point. So that's our cardiac muscle, our smooth muscle, our glands. Actually, adipose tissue might also be added to this list. So fat cells do some homeostasis as well. Sure. So if you think about like how the spine works, for example, like the beginning of this neuron, its cell body would be in the spinal cord, but then its axon would start to branch out of the spinal cord where it would become peripheral nervous system. And then this ganglion would be fully within the peripheral nervous system. You guys done the phys lab with like the reflexes and stuff? I think at least some of you looked at something sort of like this in there. I'm not totally up on what's going on in phys lab this year, but I know in the past this content comes in there a little bit. So an intrinsic neuron we're not really going to think about them, but would just be one contained fully within within this area. So we're not going to worry about them. You do want to know about is the location of our neurons that are going to go out and form our sympathetic nervous system. So their cell bodies are going to be in a very specific part of the spinal cord. So when we're looking at those preganglionic cells that are becoming the central nervous system, that are becoming the sympathetic nervous system as they branch out, they're starting in what we refer to as the thoracolumbar region. So that's all your vertebrae in the thorax, so T1 through T12, and then the first two lumbar vertebrae. So we're going to have our nerves coming out of the central nervous system from T1 to L2. And then most of those nerves are going to go into something called the sympathetic chain, which is where you'll see their ganglia. And so where they'll be forming their connections with postganglionic neurons. So usually the preganglionic neurons in the sympathetic nervous system are pretty short because the sympathetic chain actually lies right along your spinal cord. So eventually when you get into the chest cavity, probably won't be until A&P 2 because we gotta go through legs first. You'll see that we have this chain lying like a white stripe kind of right along the vertebrae. So these axons are pretty short between the central nervous system and the sympathetic chain. But they, from the sympathetic chain, send out their postganglionic neurons really far. So our postganglionic neurons will go from the sympathetic chain all the way up to the eye, go from the sympathetic chain all the way down to the genitalia. So they're branching out much farther. Our postganglionic neurons will be bigger. There are some exceptions to this idea that they're synapsing in the sympathetic chain. So we also have these things called collateral ganglia. So this is, so this is the bottom of the sympathetic chain that we see here. But here we can see a collateral ganglion lying kind of like a bead all on its own. So there are a couple ganglia that aren't part of the chain, and that's when we call them a collateral ganglia. So some of these preganglionic neurons, instead of going to the sympathetic chain, are going into the collateral ganglion and then into the effector organ. And in that case, we would expect to see potentially a slightly longer preganglionic neuron. The other exception is when we're talking about the nerves going to the adrenal glands. So your adrenal glands are going to lie on top of your kidneys. They're going to be producing a lot of things, but particularly we're interested in things like stress hormones coming from the adrenal medulla, so the inside. Okay. So for the sympathetic innervation here, we have our preganglionic neuron reach all the way into the center of the adrenal gland, where it synapses with something called a chromaffin cell, which is like a specialized super teeny tiny postganglionic neuron. So we give them their own name because they practically like don't have axons, but that's really what they are. That is basically a postganglionic neuron. So when they do this, they're going to release two molecules called catecholamines. That's just a class, basically, of neurotransmitter or hormone. They're going to be releasing epinephrine, about 80%, and then 20% norepinephrine, and then a little bit of dopamine. And that bottom picture is just showing you where the adrenal glands lie on top of the kidneys. And then it's showing you that we sliced one in half. And then it's showing you the relationship of those cells. So our sympathetic preganglionic neuron going all the way into the adrenal gland, into the central part called the medulla. And then we have our little chromatin cell releasing our epinephrine and norepinephrine. Here we picked epinephrine because there's more of it.
You don't have to have it close.
So the chromatin cell is the postganglionic cell here. So basically you do. That's why we're specifying that that's like a modified version of our postganglionic cell. Otherwise we wouldn't have a ganglia, right? We just terminate on an effector organ. So if we're using the word preganglionic, it by necessity has a postganglionic. This is just that map zoomed in bigger so we can see the three types of relationships we have in our sympathetic nervous system. So here, our main type is that we have preganglionic from the central nervous system into ganglia in the sympathetic chain. And then our postganglionic out to our effector organs. Could be things like cardiac muscle, smooth muscle, glands. We might also have a preganglionic neuron go all the way into the adrenal medulla and then form a connection with a chromatin cell, which is like our modified postganglionic neuron. Or we might just go to a collateral ganglion. So a ganglion out to the side, out by itself, and then have a slightly shorter postganglionic neuron to do that. So when we're talking about the sympathetic chain, we're actually doing a kind of complicated pathway when we jump into it. So if we look at that preganglionic neuron, so our cell body is here in the spinal cord, okay? And it's going to exit, its axon is going to exit through something called the ventral root. So ventral means like the same side of your stomach, so it's kind of coming off anteriorly. So we're leaving through this ventral root, and then we're going to jump onto a spinal nerve and into the sympathetic chain. So we're kind of looping through here. So we call this loop in the white ramus because this is going to be myelinated. Okay. So it's white in color. And the axon leaving the sympathetic chain, we're going to say leave by the gray ramus. So the gray ramus would be unmyelinated. So as we have it jump off, it's going to go back onto the spinal nerve and out to the effector organ. And you'll actually be able to see these two rami in lab when you eventually get down that deep into the body. These are actually at this point visible structures. But basically, the system is we're jumping on the spinal nerve into the sympathetic chain and then back onto the spinal nerve and out to the effector organ. It's this row of ganglia, and the reason the chain exists is that we actually don't have to stay at that same level. So this nerve could go into this ganglion and then have its connection like go up and then hop out on a different spinal nerve. So that's why they're connected, is so that we can kind of have that mixed thing going on. But yeah, each spinal nerve isn't just like one neuron, it's several neurons together. Is that what you were asking?
Yeah, I got it.
Okay, cool. So our postganglionic neuron is the start fluon, leaving the ganglion on the gray ramus and re-entering the spinal nerve. And it's going to its effector organ. The same sort of thing would happen if we had a collateral ganglion. I'm going to draw one on the bottom. Let's say now we have a collateral ganglion over here, not connected to the chain. So we'd follow the same sort of route. We'd still be starting here in the spinal cord, jump on a ventral root and onto the spinal nerve, but we wouldn't do this little loop into the sympathetic chain. We just keep going until we get to the collateral ganglion, at which point that's where we'd see our postganglionic. So it's the same process, just without this little detour. So when we talk about the parasympathetic neurons, they're going to be originating in a different place. Our sympathetics were in the thoracolumbar region, right? So basically your chest and the first part of your lower back. Our postganglionic neurons come only from the brainstem or from the sacral spinal cord. So they're really just at the top and the bottom. Sometimes people refer to them as like craniocaudal, really coming from the brain or from the very, very base of your spine. This means that they have a fair distance to travel because they have to go out to all those organs in between. So we're going to see pretty long preganglionic neurons here because their ganglia are essentially going to be right on their effector organs. So they don't go into the sympathetic chain. That's just for the sympathetic nervous system. They don't even go into their collateral ganglia. Those were for the sympathetic nervous system as well. They're going almost all the way out to where they're going. And then those ganglia are right by the effector organ. So a difference we could say between the sympathetic and parasympathetic nervous system would be like sympathetic, short, preganglionic, and parasympathetic as a long preganglionic cell. And we saw that there are some exceptions in the sympathetic nervous system, but this is a pretty good approximation. Their parasympathetic nerves coming from the brainstem are actually on very specific cranial nerves, not all of them. The one we're mainly going to see out in the body is cranial nerve 10, the vagus nerve. So the vagus nerve, we saw it in lab, but we're going to see it again as we go into the neck, as we go into the chest cavity. So you can see the course of the vagus nerve. It's starting here as a cranial nerve, brainstem. It's spreading all the way down and even giving innervation all the way as far as our small intestine. So it is traveling a long way away from where it starts. Our other cranial nerves mainly stay in the head, or at least that one exception, the spinal accessory in the upper back there. So the other three cranial nerves that carry parasympathetics, cranial nerve 3, the oculomotor nerve going towards the eye, cranial nerve 7, the facial nerve, which basically goes into the face, as its name might suggest, and cranial nerve 9, the glossopharyngeal nerve, goes a little bit into the neck. So most of our body is really receiving almost all its parasympathetics from that vagus nerve. Regarding the sacral part of the parasympathetic nervous system, it's doing some more pelvic organs, basically. The vagus nerve isn't going down all the way into your pelvis. So we have pelvic nerves coming out, and they do some large intestine, which you'll see when we get to GI. It's like the second part of your intestinal system, going to your rectum, going to your bladder, and going to your genitalia. So those sacral nerves aren't traveling anywhere near as far as the vagus nerve. The vagus nerve really carrying the team here for the parasympathetics. And this is just a note that these pelvic nerves that carry parasympathetics are separate from our somatic spinal nerves, which we'll see going to like muscles in that area. So the preganglionic neurons are coming from a brainstem. Realistically, we can think of this as probably the vagus nerve, but it might be oculomotor, facial, or glossopharyngeal, right? It's going out through the PNS, really close to its effector organ. We have this long preganglionic neuron, and those pelvic nerves are relatively long when we compare them to the sympathetic nervous system. And then we have our postganglionic nerves, our postganglionic neurons to the effector organs. And that's the part that's short here. So next we're going to talk about how these different parts of the nervous system communicate with those effector organs and how they communicate between preganglionic and postganglionic. So we're going to be focusing in on some specific neurotransmitters and some specific types of receptors. So when we say something is cholinergic, that word comes from acetylcholine. So our neurotransmitter that would go to a cholinergic receptor, acetylcholine or ACH. So anytime you see that a neuron is releasing acetylcholine, you should expect that on the other side of that synapse, we're going to have cholinergic receptors to receive that message. The opposite is the adrenergic receptors are going with norepinephrine. And the reason for that is that another word for this is noradrenaline and adrenaline. So adrenergic receptors would do both epinephrine and norepinephrine. We use the words epinephrine and norepinephrine, but it's the same thing as adrenaline. So to figure out what type of receptor we're going to have in these different parts of the autonomic nervous system, we're going to think about what type of neurotransmitter these nerves are sending out. So anytime we see a preganglionic neuron in either the sympathetic or the parasympathetic nervous system, it's going to be releasing acetylcholine. So what do you think that's going to mean for receptors on postganglionic neurons? What type would you expect to see? Check that previous slide. We have acetylcholine. What receives the signal from acetylcholine? A cholinergic receptor. Yep. So our preganglionic neurons release acetylcholine, which means our postganglionic neuron is going to have a cholinergic receptor. Then we're going to divide whether we're talking about parasympathetic or sympathetic postganglionic neurons, because they release different neurotransmitters to the effector organs. So the parasympathetic postganglionic neurons also just release acetylcholine, which doesn't feel easy now, but that actually makes the parasympathetics pretty easy in terms of neurotransmitters and receptors, because they're releasing acetylcholine from their preganglionic neuron, they're releasing acetylcholine from their postganglionic neuron. So we don't even have to worry about epinephrine, norepinephrine, adrenergic receptors. We just do acetylcholine stuff. Our sympathetic postganglionic neurons, though, are going to be releasing norepinephrine, which means that here with the sympathetics, we have to think about both types. So we're going to be releasing acetylcholine from the preganglionic neuron, and then we're going to synapse with our sympathetic postganglionic neuron, which is going to release norepinephrine at its effector organ. So then we're going to have to think about adrenergic receptors. And we have the adrenal medulla here separately because it releases both epinephrine and norepinephrine, mostly epinephrine. But this is part of the sympathetic nervous system. So you'll note norepinephrine is coming out of both, right? And then epinephrine is a similar type of molecule, so it's also going to correspond to adrenergic receptors. So basically everywhere we have an organ receiving communication from the sympathetic nervous system, that organ is going to need to have some type of adrenergic receptor. So adrenergic receptors are going to be specific to the sympathetic nervous system, basically. Everywhere else, we should be expecting cholinergic receptors. So here we see how that works. So this is looking at the sympathetic nervous system. That's the slightly more complicated one. So here we see, for example, let's pretend that this is a ganglion in our sympathetic chain. So our preganglionic neuron is releasing acetylcholine. We see a type of cholinergic receptor here. I'll talk about the different types in a second, but specifically this is going to be a nicotinic cholinergic receptor. So once we go over what that means, we can come back to this diagram. And then the postganglionic neuron, if we follow it down, it's adrenergic, it's releasing norepinephrine, which means that on the effector organ, we're going to have an adrenergic receptor. And we'll see that there are subtypes of those as well. So that same thing is happening regardless of what type of sympathetic ganglion we have, basically. So still, if we have a preganglionic neuron going to the adrenal medulla, that preganglionic neuron is still cholinergic, still releases acetylcholine. So we still need a cholinergic receptor inside the adrenal medulla on those chromatin cells. And then we release epinephrine and a little norepinephrine and have our adrenergic receptors. And then if we go into the collateral ganglia, it just looks the same as the sympathetic chain situation, except for that preganglionic neuron is a bit longer. So still acetylcholine, then a cholinergic receptor, then norepinephrine, and an adrenergic receptor.
What's the difference between the epinephrine and the
They're just slightly different molecules, and I couldn't really tell you what they look like. I would go ask a chemistry professor. I'm not even going to attempt to draw them. And actually, we'll see that a bunch of the receptors can receive either epinephrine or norepinephrine, but they have a slight preference for one or the other.
One of those is the adrenaline.
Yeah, I think the epinephrine and adrenaline. So like an EpiPen is epinephrine, which might be a good way to remember that epinephrine is with the sympathetic, right? I think an EpiPen, right, like wakes you up, I assume. I've never had anybody stick me with an EpiPen, if any of you. You don't have to tell me. But, you know. Yeah. So here's just the picture of the parasympathetic nervous system. Super nice. We've got acetylcholine from the first one, and we have a cholinergic receptor, and we have acetylcholine again, and another cholinergic receptor. Cool. So next time we'll just pick up on those two types of cholinergic receptors, and then we'll go into the different adrenergic